This is the Wnt pathway in C. elegans and Xenopus embryos. Today we'll be talking about what the Wnt pathway is and how it affects the early embryonic development of these two model organisms. Now, Wnt signaling is actually used in the development of many species and can be seen conserved throughout the multiple model organisms that we are showing today, such as C. elegans and Xenopus. Shown above is the general pathway of Wnt signaling, which begins actually with Wnt-Ligand, or some sort of ligand, binding to the frizzled receptor on the cellular membrane. This activates a protein called disheveled. Now, the activity of disheveled is actually to block the activity of GSK3 kinase in the cytoplasm. GSK3 kinase phosphorylates Β-Catenin, which marks it for degradation and also does not obviously allow it to enter the nucleus. Right? So since disheveled is activated, GSK3 kinase is repressed. That means Β-Catenin can enter the nucleus. After Β-Catenin has entered the nucleus, it can now bind to TCF, which then those two form a transcriptional activator. Without Β-Catenin in the nucleus, Groucho actually binds to TCF, and that's a transcriptional repressor. Now that I've introduced to you what the general Wnt pathway looks like, we will see in action in specific model organisms, starting with C. elegans. So this is a map of the C. elegans embryo in its early developmental stages, going from the zygote all the way to an 8-cell stage. So the important thing to note is at the 4-cell stage, there is 4 cells called ABA, ABP, P2, and EMS. And if you look at the difference between the 4-cell and 8-cell stage, the EMS cell actually divides into 2 daughter cells called MS and E. Now, if MS and E are actually allowed to develop by themselves autonomously, as if no cell-cell interaction can take place, both those cells will adopt a MS-like fate, which kind of implies that MS and E are different because of some sort of cell-cell interaction or cell-cell signaling. Now, this cell-cell signaling actually comes from the contact between P2 in the 4-cell stage and the posterior end of EMS. What's shown here is actually a flowchart of some transcriptional regulators in C. elegans, and if you can note, MED1 and 2 is actually present in the EMS cell. And interestingly enough, MED1 and 2 activates both T-box35 and End1 and 3 in MS and E respectively. Now, something must occur to where cells from the same source, which is EMS in this case, end up expressing different genes to specify different fates, which is MS versus E. So Wnt signaling actually plays a role in differentiating the MS and E cells. As you can see here, Wnt signaling is actually present from P2 to the posterior side of EMS. Now, looking at the flowchart on the right, MOM2, which is actually Wnt-Like, and MOM5, which is the frizzled receptor, are both a part of the general Wnt pathway shown previously and receive their name from their mutant phenotypes, so more of MS. Okay. So this pathway points specifically to the role of Wnt signaling in repressing the MS fate. If Wnt signaling is not present between P2 and EMS, both anterior and posterior halves of EMS will develop into MS-like cells. So the Wnt pathway is actually specifically used to signal WRM1 and LIT1 to phosphorylate POP1 protein in the nucleus of the cell. This results in transportation of phosphorylated POP1 to be transported out of the nucleus and interact with a Β-Catenin protein called Cis1. So the polarization of MS and E cells can actually be observed by the higher levels of POP1 in the MS cell versus the lower levels in the E cell due to the WRM1/LIT1 phosphorylation. Lower levels of unphosphorylated POP1 in the nucleus reduces its ability to repress E-like development and POP1/CIS1 also contributes to the activation of E-fate, determining factor N1 and 3. So now we will look at the Wnt signaling in Xenopus embryos. So for a little background, Xenopus embryos actually undergo something called cortical rotation during normal embryonic development, and this is shown above. This is when the animal cells, which is the top half, right where the sperm entry point is in the dark shaded area, actually rotate down towards the vegetal pole, which is the unshaded half of the embryo. Now this is done with the activity of microtubules, and if the activity of microtubules is disrupted by the application of UV light, cortical rotation will actually not occur. Interestingly enough, when cortical rotation does not occur, the embryo does not develop any dorsal structures and is ventralized. So evidence for the role of the Wnt pathway in developing dorsal structures in the Xenopus embryo is actually shown above. Right? If you inject Wnt or disheveled or Β-Catenin or dominant negative GSK3 mRNA into the future ventral site, it will actually cause the development of a secondary dorsal axis, which is shown to the right. And you can see how this Xenopus embryo is actually contains two dorsal structures, which is the head, eyes, neural tube, stuff like that. And also, the hyperdorsalization caused by treating embryos with lithium chloride also has ectopic nuclear localization of Β-Catenin. Now, this is important because if you recall the Wnt pathway, Β-Catenin needs to be able to enter the nucleus to interact with TCF and create a transcriptional active. So what does cortical rotation actually have to do with specifying dorsal structures through the Wnt pathway? So looking at the first picture on the left, the shell of protein is actually contained in vesicles located at the vegetal pole. Now it's important to note that this disheveled protein is actually not zygotic in nature, and is actually deposited there by the mother. Now if we recall, disheveled in the Wnt pathway is an anti-GSK3 protein that prevents GSK3 from phosphorylating Β-Catenin to initiate its degradation. Now, during cortical rotation, these vesicles containing the disheveled protein are actually moved to the future dorsal side of the embryo and is able to stabilize Β-Catenin at the future dorsal side. So this image above actually shows a couple of stages after cortical rotation has actually occurred. And as you can see, Β-Catenin is actually established in a gradient with the highest concentration at the future dorsal side. Maternal Veg-T and Veg-1 is also present in a gradient at the vegetal pole of the embryo. The overlap of Β-Catenin and Veg-T and Veg-1 creates an area of high nodal-related protein expression. Now, high nodal-related protein expression specifies the Nieuwkoop center, which induces the Spearman organizer just above it in the future dorsal side. So just to recap, Β-Catenin with Veg-T and Veg-1 specifies a high level of nodal-related proteins, which then goes along to specify the organizer, whereas Veg-T and Veg-1 by themselves without Β-Catenin specifies only a low amount of nodal-related proteins, and that goes on to specify the ventral mesoderm. Now, the Spearman organizer is required for the proper development of dorsal structures and also interacts with the Wnt pathway, albeit in a different manner than what has been shown so far. So stage 8 and stage 9 actually show what we have already gone over, which is high levels of nuclear Β-Catenin with Veg-T Veg-1 specify a high level of nodal-related proteins. Right? The area where the concentration of nodal-related protein is actually the highest specifies the Nieuwkoop center, which then goes on to induce the organizer in the future dorsal side. Now, interestingly enough, looking at stage 10, the organizer actually expresses Wnt antagonists, which is Frisbee, right, and also BMP antagonists, right. Now, this can seem a little contradictory, but it can be explained by the timing of events in the developing Xenopus embryo. So in the early embryo, before gastrulation, the specification of the organizer requires the Wnt pathway to stabilize beta-catenin, but after the mid-blastula stage, when the organizer has already been specified, a zygotic expression of Wnt proteins, in this case X-Wnt8, is used to specify ventral mesoderm. To ensure the proper development of dorsal mesoderm, the organizer secretes Wnt or BMP antagonists. In the Xenopus development, the Wnt pathway is used to solve problems of specification in multiple different situations and at multiple different times. This is also seen widely in the development of other species as well. So Xenopus embryos also use Wnt signaling to properly specify the dorsal dermamyotome domain of the somite, which goes on to become dermis and trunk muscle. So this is another example of how the Wnt pathway is used to solve a different issue other than what we have seen before. So to sum up, the Wnt pathway in C. elegans and Xenopus embryos is used in cell-cell signaling to differentiate cells and induce the development of structures that would otherwise not be specified for if these cells were allowed to develop autonomously. So it's important to keep in mind that the Wnt pathway can be used at different times and at different locations to ensure proper development. And the examples that were shown here are just a small sample of what it can be used for. So to check your understanding of the topic, go ahead and diagram how Wnt signaling is used in the development of C. elegans and Xenopus embryos, and ask yourself what's similar between the two and what's also different.